Good afternoon. I hope everybody's enjoying this year's ALS Association Greater New York Chapter's Educational Summit. Now's the time for you to buckle your seatbelts because the next 90 minutes promises to be filled with exciting new information about current, ongoing, and even futuristic research that is very exciting for our ALS community. We are about to hear from Dr. Jinsy Andrews, who currently serves as the Director of Neuromuscular Clinical Trials and is an Associate Professor of Neurology at Columbia University. In addition to overseeing clinical trials, she also serves as a doctor for many ALS patients at Columbia. She is the co-chair of the Northeast ALS Consortium, we all know as NEALS, which is a network of over 100 clinical centers. She is a fellow in the Academy of Neurology and serves on the Academy Neurology of Science Committee. She got her BS from Union College, her MS from Columbia's Mailman School of Public Health, and her MD from Albany Medical Center. She did her fellowship at Columbia University, and she is board certified in neurology, neuromuscular disease, and electric diagnostic medicine. We're also going to hear from Dr. Noam Harel, who is a neurologist with experience in both basic and clinical research. He completed his undergraduate and graduate degrees at the University of Pennsylvania, and then his neurology residency at Columbia University. He is now an associate professor in neurology and rehabilitation and human performance at the Icahn School of Medicine. He is a principal investigator at the Spinal Cord Damage Research Center at the James J. Peters VA Medical Center in New York. His primary interests are in rehabilitation from and treatment of spinal cord injury. He's also interested in ALS. His clinical research lab, located at the Bronx VA, uses targeted physical exercises, electronic stimulation, ischemic conditioning, and other methods to reactivate weakened nerve function. He is funded by the VA, the NIH, New York State, and the Craig Nelson Foundation. He is the chair of the American Academy of New York. He is a chair of the American Academy of Neurology Spine Section. He's on the board of directors of the American Society of Neurorehabilitation and on the editorial board of the Journal of Neurology. Finally, we're going to hear from Kurt Hagstrom, the Chief Commercial Officer at Sycron. He's a marketing and business development executive with extensive experience in strategic marketing, sales leadership, business development, and R&D engineering. He has a proven track record for developing and launching products that drive top-line revenue. Sycron is at the front line of ALS technical research. Sycron has developed the first brain-computer interface system. What is that, you may ask? A brain-computer interface system is a permanent stent-like implant designed to enable patients to wirelessly control digital devices through thought and to improve functional independence for people who are paralyzed. Sycron's research found that 1 year after the device's implantation, none of its 4 participants had experienced any adverse events that led to further disability or death. Nonverbal ALS patients and their families are very excited to see this system coming to fruition. Thank you for listening and enjoy your research ride. Jinsy, take it away.
Thank you. Thank you very much for that kind introduction and the opportunity to speak at this very educational summit, which I think is important because so much is happening in both the clinical side and the research side for ALS. It seems like it's so tough to keep up. So I will take the opportunity today to start talking about the evolving landscape in ALS and really talk about the research updates that have started to kind of come to a head really in the past year. So let's see, I'm just going to try to get us up here. Slideshow. All right, here we go. So as Teresa said, I would put your seatbelts on because it's going to be pretty exciting and a very thorough review. So what I'll do first is, I know this is a research and advancement update, but I wanted to touch a little bit about the current treatment options for ALS, because we're learning about them as we go. And there is research that's going even after something gets approved by the FDA. So first, I wanted to highlight Riluzole. So we know about Riluzole. It used to be called Rilutech as a brand, and it's been around for more than 20 years, and the trials occurred in the 90s. And we know now that in more recent years, there's a liquid formulation and a film formulation that was brought to market. And during that period, I think there were a couple of key things that the community needed to know about whether we needed to get new formulations or just move on to other new drugs and do clinical trials on other investigational or experimental therapies. And so when revisiting Riluzole, I'll just stop here for a minute to reflect on a couple of key things. Number one, there was a publication out of the UK that went back and looked at this clinical trial data, the original clinical trial data from Riluzole in the 90s, and tried to categorize patients in stages. And what they were able to see is that there was a delay from moving from one stage to another. And so that was a retrospective review. And one of the limitations in that study was that back in the early 90s, when we enrolled in those original clinical trials, individuals were in stage 2 according to the King's staging criteria, or later when they entered into the original clinical trials. So we didn't have clear information of whether starting Riluzole in stage 1 or early stage ALS would be beneficial. But subsequently, a couple of more key publications have come out. So in 2020, there was a publication, which I was a part of, that looked at population-based databases across the globe and tried to look at whether Riluzole had an advantage, people taking Riluzole had an advantage in survival compared to those that weren't post-approval. And so in this retrospective study, the survival and advantage was anywhere from 6 to 19 months compared to those that never took Riluzole. So the benefit was more in the real world evidence than what we saw in the original clinical trials. But most recently, Dr. Mitsumoto published a paper looking at a clinical trial database called PROACT. And within that database, they looked at Riluzole use and survival in people who were taking it within 6 months, 12 months, and later. And what they found that in individuals that had early diagnosis and started Riluzole early, they had better survival than those that were diagnosed later. And so it seems to become more important that starting therapies to slow down disease progression are very critical to be started as early as possible in the disease journey. And of course, in 2017, we got the intravenous formulation of edaravone. In May of last year, 2022, we got the oral form of edaravone. And I just put this here because you'll hear it in other presentations, but I think it's good to reiterate that the dosing administration is similar between the intravenous formulation and the oral formulation. The doses are slightly different, but they're considered what we call bioequivalent. So they create equal exposures in your body and your system. And so the studies that were done, or the research studies that were done to kind of bring that oral form into market, was really to look at whether the exposures from the oral drug edaravone was similar to those that were taking intravenous drug. And last, everyone's familiar with Relyvrio. Relyvrio was approved based on a phase 2 study. It was a 6-month study, placebo-controlled, that looked at treated patients versus placebo patients. And in this study, there was a statistically significant effect on a pre-specified primary endpoint. And then subsequent information from the open label extension, which showed a survival benefit, helped provide the supportive confirmatory evidence for its full approval in the US. And so this drug is an oral drug that is in a powder pocket. typically has to be mixed in 8 ounces of room temperature water. And then initially people will take it 1 time a day. And then after 21 days, they would take it twice a day. So knowing that Riluzole, edaravone, and Relivrio are now available in the US, this is a research talk. Why am I talking about it? Because it's important to talk about it with your healthcare provider and really make decisions about whether you want to take it before considering a clinical trial, because typically in a clinical trial, you're restricted from starting any of those therapies after you've enrolled. So it's ideal to consider those medications, decide whether you want to take it or not with your healthcare team. And then you can always layer the clinical trial and the investigational or experimental drug on top of what we call standard of care. Now, just in the last week, we got news about a new gene-targeted therapy that's available on the market. So genetics in ALS really represents a smaller portion of all people with ALS. So 90 to 95% are what we consider sporadic with no gene mutation that we can identify today. That may change in the future. But the 5 to 10% will have some associated genetic mutation. The list of genes that are associated with ALS are growing exponentially each year. But why am I highlighting this? Well, it's really important to know that there are multiple clinical trials today that are targeting various gene mutations and are actively recruiting. So knowing the genetic information, if you're at risk of carrying, or if you have a family history of ALS, is important because there might be experimental therapies that may be available should you choose to want to participate. And so one such therapy that was tried in clinical trials was tofersen. This is an antisense oligonucleotide. So essentially it's a medication, or now it's a medication that's FDA approved through accelerated approval, but it's intended to target a SOD1 gene mutation and reduce the production of what we think is toxic SOD protein. And in doing so, we hope that we can preserve the motor neurons. So it's delivered through a spinal tap, and it is something that requires you to go to a hospital or a facility to get the drug administered. What happened in this trial? So we treated patients. There was a 6-month placebo-controlled study. Columbia was one of the participating sites. I was an investigator. And what we noticed is that people who were treated with tofersen had, and that's the red line, they had lowering of the SOD1 protein in their spinal fluid. They also had lowering of a marker called neurofilament. This marker is a marker of axonal injury or nerve injury. So we were able to reduce that with that drug. And then the placebo was the blue arms. Based on the neurofilament markers, we were able to achieve what we call accelerated approval. Now, aside from the neurofilament markers, there was a signal that was consistent across ALS functional rating scale, slow vital capacity, muscle measurements when you do handheld dynamometry, and survival. So the data was all consistent across all measures that were taken during the clinical trial. And so this all led to what we call an accelerated approval, which is different from a full approval, but the drug brand name is called Qalsody. And if you are someone with ALS and have an SOD1 gene identified, it's important to talk to your healthcare provider about whether this gene therapy would be appropriate for you or not, and see how to do it. It's all in progress right now. Everything is still the infrastructure is still building. So we're hoping to get that up and running in our ALS clinics across the country. So having this gene therapy on the market has had a massive impact on our research landscape, also on our clinical practice. And so for one, pre-symptomatic ALS has, especially those that are carrying SOD1, has been a target of possible treatment and prevention of ALS. So in this slide, what I'm trying to highlight is the fact that in this publication by Michael Benatar, he saw that within 12 months prior to somebody getting their symptoms of ALS, if they're carrying the SOD1 mutation, they have an elevation of that neurofilament, or that marker that I told you about axonal injury. So based on that, we were able to kind of crudely calculate a potential threshold where people are at increased risk of developing ALS if they carry the SOD1 gene mutation that is considered to be associated with ALS. And so in this study, so this is the first ever study to prevent ALS from occurring. In this study, basically patients will be tested for the gene, and if they carry the gene, they'll be followed on a monthly basis for neurofilament. And if they cross a threshold, they'll then be randomized to treatment or placebo. And remember, these are individuals with a gene carrier that do not have any symptoms of ALS. They'll be treated, and if they develop symptoms, they then get automatically treated with tofersen. If someone's being followed and they never cross the neurofilament threshold and they develop symptoms, because we're not entirely 100% sure that that's the right threshold, they'll then be shuttled to treatment in Part D, so that anyone who develops ALS across the study will end up getting treatment with tofersen now that it's approved. So Columbia is one of the participating sites for this study testing tofersen in pre-symptomatic individuals with SOD1. Now, the clinical trial landscape, it is big, and I only have so much time to explain it to you. So the first is that this should show you kind of a big picture of all the clinical trials that are occurring in the US, and the bolded ones are the ones that kind of came on to the scene this year. So baricitinib is something that's targeting C9orf72, and it's a small phase 1 study, but interestingly using what's called a basket design. So essentially there's the same outcome for everybody. So the outcome measure is the same, but in a basket trial, you're looking at different diseases that you can measure the same thing in. So in the baricitinib study, we're looking at frontotemporal dementia and groups with ALS, and then measuring a protein that occurs when you have the C9 mutation. QRL201 is an antisense oligonucleotide targeting STAFMN2 and is actively recruiting. AP101 is a human monoclonal antibody targeting SOD1, and they are recruiting sporadic ALS as well as those with the SOD1 mutation. BLZ945 is an investigational or experimental therapy targeting colony stimulating factor. It's supposed to reduce activation of macrophages, which is thought to be related to inflammation. Dazucorilant, Columbia is hoping to be a site for this, but it's a investigational product that's targeting glucocorticoid receptors. And then PTC Therapeutics also has a therapy that's starting to recruit, as well as Sanofi with SAR443820, which is a RIPK1 inhibitor and also targeting neuroinflammation. And then one big news is that one of the phase 3 drugs, reldesemtiv, which targets skeletal muscle activation, was actually stopped for futility, and that announcement came last month as well. Now, when I talk about cell, I used to keep all these on one slide, but I've actually had to separate it out because cell therapies have grown tremendously in the ALS field. So when I say cell therapy, it confuses people because some people think automatically stem cells, but there's different types of cell therapy. And actually, as our field matures, we should really be careful about when we say stem cells, what type of cells are they and where are they derived from? So cell therapies on this slide is a combination of both T cell therapies and stem cell therapies. And so allogenic T cell therapy is being tested in ALS and is actively recruiting. I'll go over that in a minute. Autologous T cell therapies are being offered through Rapid Therapeutics and Koya Therapeutics. And then I think the most popular question I get is Brainstorm and Neuron, which is autologous. So it's bone marrow derived from your own body, stem cells that are then cultured and then engineered to secrete growth factors and then infused into the spinal fluid through a spinal tap. and that completed a phase 3, and FDA announced that there will be an advisory committee, although a date has not been set to discuss the clinical trial data results. And then the Mayo Clinic continues. They are not enrolling any more patients, but they are evaluating adipose tissue or fat cells that can be turned into stem cells and then infused into the spinal fluid. And then Cedars-Sinai is also injecting stem cells directly into the brains or motor cortex of people living with ALS. The T-cell therapy, the allogenic T-cell therapy, is something that's being conducted at Columbia University. So Dr. Schneider is leading that study there. So the thought is that T-cells are related to the neuroinflammation that makes the motor neurons sick. And so if we can put healthy T cells through intravenous infusions into the body, we can slow down disease progression. And this is the eligibility criteria. The toughest criteria to meet in the study is that ALS-FRS score between 36 and 45. So in parallel to the stem cell studies, the T cell studies, there's also gene-targeted therapies that are in clinical trials. So there's a variety of platforms. So I talked about antisense oligonucleotides, but there's also viral vector-mediated gene silencing techniques or gene delivery techniques that are actually in preclinical studies right now for ALS. And CRISPR hasn't quite made it there yet, but other ways of targeting therapy include monoclonal antibodies and other small molecule approaches that can downregulate a particular protein that we think is not good for the motor nerves. So there is AP101, which is targeting SOD1, as I mentioned before, TPN101, AL001, which is another monoclonal antibody. And then there's PIK5 kinase inhibition, as well as antisense oligonucleotides targeting specific genetic mutations. The one that Columbia is leading is the FUS antisense oligonucleotide development. So this is a phase 3 study, complicated study, led by Dr. Schneider, that is recruiting any individual with a confirmed genetic mutation in the FUS gene. So again, when I'm talking about gene-targeted therapies, it's important to get screened for genetic mutations. These mutations must be reviewed and approved by a variant classification committee. And then there's additional criteria that can be a little tricky to meet inclusion, but it's intended to slow down disease progression in FUS-associated ALS. In addition, Columbia is also working with NLORMs and Target ALS in trying to do targeted antisense oligonucleotide aiming at gene mutations that are extremely rare. And so it requires giving blood samples and skin samples and having NLORM evaluate the mutation and see if they can help manufacture the antisense oligonucleotide specific to that specific rare gene mutation. Finally, I'll focus on the platform trial. It's already many people are already familiar with it, but essentially the platform trial in ALS has launched in 2020, and within 3 years we were able to get answers for 4 different drugs. So regimen A, which was the Zilucoplan, stopped for futility. Regimen B was a drug that did not show any effect on primary or secondary endpoints. Regimen C, which was CNM-AU8, it's gold nanoparticles to help with the bioenergetics of the cell, that did not hit its primary or secondary endpoint, but there was a question of a particular dose, the 30 milligram dose, having a benefit on survival. So likely it will need a confirmatory study. So it helped us pick out a dose and an outcome for our confirmatory study. And regimen D did have some signals here. So regimen D is pridopidine, and this is a sigma-1 receptor agonist. And essentially, it did not hit its primary or secondary endpoints, but there was this pre-specified analysis of quantitative speech measures, which did seem to benefit while taking pridopidine. But in a post-hoc analysis, if you kind of look at the cohort of people that were rapidly declining, there was an effect compared to placebo, and that was associated with a reduction in neurofilament. So likely that will also move to confirmatory studies. And then as we read out the first 4, the next 2 are launching. So ABBV CLS 7262, wish it had a name, but I'll call it regimen F for now. And regimen G, DNL 343, are both small molecules that can affect the integrated stress response and try to reduce the toxic proteins that lead to injury to the motor neurons. So that's coming in the platform. And this is just a picture of the sites. So in the New York area, we have a couple of sites that participate in the platform trial, but those regimens are also coming. And last, I think I'll just focus on, and this is a little segue to the next speakers, but while we're trying to find better gene therapies and better therapies to slow down disease progression, we also want to improve quality of living in ALS. And so at Columbia, we've been working on head drop, which is a problem for some people with ALS, but it really does affect people's ability to swallow, to breathe, and also to socially interact. And all of the things that we use today are static neck braces that really don't allow for much head movement. And so the engineering students there have developed these cool joints that can help you move in rotational ways. And they applied it to a neck brace or neck support system. And so you can flex, rotate, and tilt your head in the system. And if you have no strength at all, you can use tracking devices like joystick or eye trackers to help move the head for you, or you could just provide passive support. But essentially, all the hardware that you see is in the back, but in the front, there is nothing to impede someone's swallowing or breathing with constriction on the front of the neck. So interestingly, the parts are 3D printed. So it's hopeful that we can get more data and try to see if that's something that could be commercialized in the future. So I'll just leave with saying, I always leave with this slide because I want to encourage kind of learning. When you do clinical trials, it's not just about the experimental therapies that people try to access, but when you participate in a clinical trial, if you give your sample, or if you do a survey, or you provide your clinical information, it helps us learn about the disease. And that's research too. So whether you get access to a device or an experimental therapy, it doesn't matter. Whatever you do in research helps the cause to find a cure for ALS and try to find ways to live better with ALS. But we still need basic research. We still need to translate more genetic and cell therapies into clinic. While we're doing these research, we have to continue to do biomarker discovery, and we still want to include kind of innovative ways to evaluate things. So doing exploratory endpoints, self-reported patient outcomes, adaptive trials, using AI and prediction algorithms. There's all the wave of the future. So there's more to come, and likely a lot more to say next time we have an education summit. But for now, I'll turn it over to my colleagues to continue the conversation about some of the technologies in ALS that we're able to use. So with that, I'll turn it over to Teresa to introduce our next speakers.
Okay, I think we're going to have Kurt Hagstrom start, correct?
That's correct, yes.
All right, Kurt, take it away. Thank you.
Thank you. And Dr. Andrews, that was an amazing talk, and it really is amazing to see all the research that's going on. And first, I just wanted to say thank you for ALS Greater New York chapter for a wonderful event last weekend with a walk. It was just a beautiful day, beautiful event, and it was great to see all the folks there. So I'm just to reintroduce myself, I'm a representative from Synchron. We're developing a brain-computer interface. And if Dr. Varela, if I don't know if you want to bring up the presentation.
You can't see it right now?
This was your slides.
Can you see it?
No, not yet.
Hold on.
And so, oh, there we go, perfect. And so I'm here today to educate everyone on a new brain-computer interface called the Synchron Stentrode that's in research right now. So if you want to move forward with the slide. Thank you. And so for what we'll be talking about today is starting to understand the concept of brain-computer interfaces. How do they work, and what functions do they have? The approach that we're taking is a pretty novel approach to how this is implanted. And we'll talk a little bit about the minimally invasive called endovascular approach to implanting this implantable BCI. Lastly, we'll talk a little bit about a command trial. So this is a research trial, and talk about what we have. It's being held actually right in one of the sites right here in New York at Mount Sinai. But wanted to educate a little bit about who's suitable for this, what's the study all about, and then Dr. Rell will talk to also about some other research that he's conducting. So just some disclosures. As I mentioned, I do represent, I'm here from Synchron. I'm actually engineer by trade, so I love working with the data scientists and the engineers to help create this technology. So it's a very exciting time. Dr. Rell has no conflict of interest with the talk itself. And also want to just make a note that the devices and procedures described in the presentation are investigational when it comes to the FDA. Again, we're not approved by the FDA, but we're in a research phase that we're excited about. Okay, go one more. So, if you look at what BCIs are, they're really the connection of the brain itself. If you want to go one more slide. Thank you. It's really the direct connection to the brain itself through the external device into a computer itself. So there's many applications that we see with a BCI. Today, we are looking at replacing the function, and it's really the function of the motor neuron, where the motor neuron can't transmit that signal anymore to the muscles, that this now BCI is able to transmit a signal to be able to control the digital environment. I think in the future, I think there's good possibilities and real possibilities to improve function and potentially even restore function as you look at these type of devices. Next slide. So many think that, brain-computer interface. So what's another terminology for this? And this is something that through the collaboration with the FDA of defining these type of devices as motor neuroprosthetics. So, and I think that's important because there is a restoration element to these devices. So it's really designed, again, as I talked about, to take the brain signals and control them and use those to control the external device. So you can see the depiction here where there's a device that places within the brain, within the sinus of the brain. So it's a blood vessel above the brain itself that reads the signals and then transmits the signals down through the blood vessels via a lead or wire and then out into the environment. So it's a wireless environment, and that goes out to whether it's a computer, whether it's a smart home environment or a smartphone. So it has many applications with that signal. And so when you look at, again, when I talked about, there's been many approaches to the BCI itself. I think there's been a long lineage of work that's been done through the craniotomy, so open surgery approach. And our CEO, Dr. Tom Oxley, he's an interventional neuroradiologist, and so neurologist. And he had thought of the idea of, because he's doing intervention on stroke, that, hey, could we do a BCI application through the blood vessels and through this endovascular approach and a minimally invasive approach, knowing that the history and the safety profile around putting stents inside the blood vessel is very well known from the cardiology world, from the peripheral world, and also the neurovascular world as of the last 10 or 15 years. And so that was his approach, and said, hey, can we do this successfully with BCI? If you want to go one more slide. So here is our stentrode. This is the very tip that rests inside the brain itself. And so you can see here, it looks just like a metal stent that you maybe seen in the past. And you can see those dots on there. Those are the electrodes that actually sense the signals through the blood vessel itself. And next, hopefully the animation will play for you. You can kind of see where this is placed within the brain and how the signals actually start to form. So you can start to see the signals in a particular part of the brain. You can see the lead that goes through the blood vessel and down the side of the brain, and that ultimately comes down through the neck and into the chest region, where there's a little device that sits just like a pacemaker under the skin that allows that signal transmission. We want to go one more slide, although we're playing again. And so when you look at the stentrode procedure, as I spoke about, it is a minimally invasive procedure, but there are some steps to it. So when we look at this, it's important, especially in our clinical study today, the early feasibility trial, that there's that pre-planning. We want to make sure that we understand where the location is in particular patients, although it's, this particular part of the brain tends to be the same in most people, but we want to make sure there's an exact measuring and markers and targets that when we go place the device, we know exactly where we're at. So there is that kind of pre-case planning. You have to make sure that your signals and things like that and your architecture of your brain is correct for the implant itself. Next, there's the deployment of it. So it does go into general anesthesia. There's a small incision in the neck here where you do the endovascular approach. And again, it's we have the neurosurgeons that are able to place the device. And then there's a little bit of a tunneling procedure that now puts it under the subcutaneous tissue right here in the chest. So these are all skills that, luckily, these are all skills that these physicians do every single day. So again, there's no really new procedures. There's no robots that need to implant the device. This is, with the hands of skilled physicians that are really across the country, but especially here in New York. And lastly is the connection piece. So once you do have the implant, this is where you start to connect and start to control your digital environment. So, whether it's a smartphone, whether it's a computer, certainly we have a protocol now to start to exercise and become BCI literate is how we're using kind of the terms is, it's something different. So there is a training element, but usually within the first training session that you do with the field clinical engineers, you're able to control the device to some degree and start to use it. So it's exciting to see that. And that's at home as well, and this is where the study is being conducted. So we're excited about that. So here you can see a little bit more detail about the system and exactly where inside the brain itself that it rests. You can see on the left-hand upper right, there's a place in the brain where we want to read the signals in which we tell you to kind of train on those. So those signals are recorded. And then again, as we talked about, you can see from the animation kind of transmits down to that external piece. And then right now, the system that we have, there's just a little bit of a paddle that rests on top of the skin. And that connects the signal that you're making through the brain signal out to the laptop, to the tablet, to the iPhone, and or the smart environment. So there's that control. In the future, this will be all wireless, but today there's just that paddle that sits on there. But again, the concept is very much similar, and again, able to connect to those different end-use devices. So now, Dr. Rao, I'd love to pass it off to you to talk a little bit more about what's here and now that, BCI and the clinical trial, and this is our command trial. So I'd love to pass it off to you.
Sorry about that delay there. You guys can still see the presentation?
Yes. Yeah.
Okay, great. So thank you, Kurt, and thank you everyone at ALS Association. It's a great opportunity to speak here today. I wanted to emphasize something that Kurt mentioned a couple of slides ago. This is not a new procedure. This is all derived from placing stents in people's brains, mostly who have had stroke and other types of reasons to deploy stents to keep vessels open in the brain. That's a known procedure that the people who are deploying the stentrode do every day for other reasons. So the new thing is actually reading brain waves with this device, but it's not necessarily a brand new procedure. So that's a very important point that Kurt mentioned. So we'll talk about the progress so far with this in humans. So the first deployment of this device was in Australia. It's not here. It's called the Switch trial that ran in Australia from 2019 to 2022. And 4 people with ALS completed the implantation and the follow-up of 1 year post-implantation of this device. Very importantly, safety was the most important or primary outcome, and there were no device-related serious adverse events at any point during the procedure or during the follow-up period. And of course, with the device, other things were, outcomes were measured, such as the ability to control a computer, as Kurt was discussing. So all 4 participants gained the ability to control a computer or a smartphone. They were able to text using WhatsApp. They were able to log in to a shopping site and fill a cart and check out. They were able to check their banking information online. So that's pretty exciting. And this is just a testimony from one of the participants in Australia who received the Stentrode as part of that study. And I'll kind of highlight the important point. It enables me to use emails and to use apps such as WhatsApp to scroll through various sites, do online banking, and whatever you can do on a computer, I can do, was, I thought, the most important point. He's a bit slower than someone without ALS would be, but he has the ability to do almost anything if he wants to. Okay, so that's, of course, the goal. I should emphasize, this device is not treating the disease itself. It is, at this point, meant to replace function. Of course, in the future, hopefully we'll develop the ability to restore and improve function as well. Okay, so the current study here in America is called the COMMAND study. Started last year. And again, motor neuroprosthesis is just a synonym for brain-computer interface. It's an investigational device. You can't just go to your local clinic and get this. It's only occurring at 3 investigational sites. All 3 sites have approval from their institutional review boards. And the FDA, of course, is overseeing the study as a whole and has approved this as a clinical trial. Okay, so this study is enrolling 6 people who have severe paralysis of the arms and hands, who have not responded to other therapies. And it's occurring at 3 different sites, as Kurt mentioned: Mount Sinai here in New York, University of Pittsburgh, and more recently, the University of Buffalo are participating. By the way, so far, 4 people have received the implant, and so we're still recruiting. And again, the main purpose of this study is to obtain more evidence of device safety and feasibility of implanting it in people, in humans. Okay. Of course, other things are being measured, such as the ability to do more online, more digital tasks, whether it's texting or emailing, finance, and of course, shopping. Okay. So hopefully this movie works with the audio. This is a person who is locked in with ALS. He has some use of his eyes, but is not using his eyes in this video. He is selecting, as you see on the right, on the zoom in of the phone, from a menu of options as he generates his communication, all of his thoughts. Can you guys hear the audio?
Oosh, that was nice.
He has a very limited ability to smile. I don't know if you caught it there, but he did smile at the end. This person is in New York. I should mention he's eager to communicate with other people who are considering this study as well. He would be happy to communicate with you. So he is able to use his thoughts to pick from a menu of items online. I should emphasize the Stentrode at this point, no brain computer interface at this point, is actually reading the mind in fluent English. He's not saying, oh, I want to order, a pizza with extra cheese and mushrooms. He's picking from items that are in front of him on a menu, a little bit similar to eye gaze, but with the thoughts. So what are the criteria? Every research study that Jinsey mentioned, or this study, or other studies that we're running in the Bronx, every study has its own criteria for entry. It can be a frustrating process. The good news is, for this study, generally accepts people who have ALS at more advanced stages than is possible for some of the drug studies. Okay, so anyone between the ages of 21 to 75 who has severe paralysis for 6 or more months, basically that means ALS, you must still have some ability to communicate, whether it is by using your voice or with an eye gaze system. We need to be able to communicate with you directly, and you directly would need to consent or not to the study. And you must have a caregiver who can help with some of the logistics of the study visits. Because this is in a blood vessel, we do screen very closely for people who have a history of blood clots in their lungs and their limbs, and that may disqualify someone. And again, primary outcome, safety, feasibility, but we're measuring other things such as you just saw, the ability to control a digital device, and we're tracking very detailed diaries from the caregivers and from the people who receive the implants themselves in terms of quality of life. And this is just an overview of the journey for people who are interested in this study. Pre-implantation, there's a relatively prolonged preparation phase, up to 3 months of extensive evaluation to make sure that someone is suitable and safe to receive this implant. Most of the visits throughout the study are at home. During that preparatory phase, there would be 2 hospital visits for specialized imaging tests, but everything else is at home. The anti-clotting medication is not so specialized. It's a combination of aspirin and Plavix, which many of you may be familiar with. There are 2 different drugs that can keep your platelets from being too active. The procedure is done in the hospital, of course, but it's a short stay. It's generally a 2-day stay in the hospital to receive it, sometimes 3 days. And then you're followed for a year, intensely followed, mostly at home. There are training visits and checkup visits throughout the year, and only 2 more hospital visits at 3 and 12 months to just get follow-up scans to make sure the device and the vessels are clean. And so far, everyone who's received the device has had clean results at all of these scans. Okay, this is obviously a collaboration between the Synchron company and the schools that I mentioned, Mount Sinai, University of Pittsburgh, and University of Buffalo. Here at Mount Sinai, Dr. Sawyer is the main contact person. So this is her email, and I'm happy to provide her and my contact information later as well. I'm just going to very briefly switch gears and talk about other research we have going on up at the Bronx VA, where I'm mostly am based. And this is a non-invasive study. So we're switching gears. This is not a brain-computer interface. What we do at our research center in the Bronx is stimulate the nervous system. And I'm going to talk about one of our studies where we use brain and spinal cord stimulation in a synchronized fashion to try to increase nerve transmission to the hand muscles. And it's all based on the premise of fire together, wire together. And that's just an easy way to describe a neural principle, a neuroscientific principle. If one neuron repeatedly fires or triggers a second neuron, so neuron A is repeatedly triggering neuron B, then the efficiency of A firing B will increase. So here, this yellow circle represents the connection between 2 neurons, or the synapse. And if we repeatedly fire A, then B, then A, then B, then A, then B, over and over in synchrony, eventually that synapse can strengthen. So that's called fire together, wire together, or Hebbian plasticity, or long-term potentiation. And this can happen anywhere in the nervous system. But what if A is the part of your brain that controls your hands? And what if B is a part of your spinal cord that controls your hands? So your spinal cord in the neck, or cervical spinal cord, controls your hands. What if we fire the part of the brain that controls your hands and the part of the spinal cord that controls your hands in synchrony over and over? Can we strengthen the connection from your brain to your hands? And again, this is all done non-invasively. That's just a video showing the types of responses we can get in the arm and hand and finger muscles from spinal cord stimulation. Can we strengthen that response? Okay. Oh, so this is just another schematic, again, showing brain, spinal cord stimulation. We individualize this. We have to measure each person's unique conduction time so that we can synchronize these machines. And then we fire in synchrony, brain and spinal cord, over and over again. I'll play that again, over and over again. And we're going to look at whether or not function and nerve transmission to the hand can improve. So that circle got a little bigger. We're tracking very sophisticated outcome measures of nerve transmission to the hand and arm. And we're tracking temporary changes in strength and dexterity in the hands for temporarily after these procedures. Just like with the brain-computer interface, this is not going to change the overall course of the disease, unfortunately. We're looking to temporarily improve nerve transmission. Okay, and so another name for this would be called spinal cord associative plasticity. So we're still seeking volunteers for this. It's anyone with ALS up to 80 years old. And again, this is not invasive. There's no surgery. And it's synchronized stimulation. So the QR code for our center's website is right there. That's my personal or my work email address. And again, we are a Spinal Cord Injury Research Center, but ALS, by the way, is partly a spinal cord disease. So we study ALS there as well. I think I'm going to stop there. I believe there already are some questions in the chat, so we should take some questions.
Terrific, everybody. Thank you so much for your time, and what a wonderful presentation you've all made for us. I'm going to start with a couple of questions that came in for Dr. Andrews. And the first question is there a surrogate biomarker like ALS-FRS that can be utilized in pre-symptomatic people with SOD1 mutation so they don't have to endure monthly LPs to monitor the neurofilaments? Or can machine learning be utilized to predict the best start time? I didn't make that question. That's somebody
That's
else.
a long question. All right, so here we go. In the pre-symptomatic study for SOD1 carriers, in that study, we're following monthly neurofilament measured in the blood. So it's a monthly blood draw. And the actual monitoring period is done with a home nursing network that goes out to the home wherever they live to draw the monthly labs. And the visits are done by video or remote visits. Just the initial visit needs to be done in the clinical center because there's like assessments that need to be done in person, including an EMG and muscle testing and breathing testing. But after that, it's just essentially all remote monitoring. It's only if you cross a certain threshold or you develop symptoms, you have to then go into the center. And if it does look like you crossed the threshold and you don't have symptoms, or you have symptoms and it looks like you're developing ALS, then you'll need to go to the hospital or the clinical center and get the lumbar punctures to get the infusion of the tofersen or the study drug, the blinded drug.
Okay, so no spinal taps prior.
Yeah, so it's not monthly. Yeah, it's not monthly spinal taps.
Okay. The next one for you is there any early indication or results in ATLAS?
That's a good question, mainly because the study has been running for quite some time now. What the major challenge is actually getting qualified participants. So these individuals have to be willing to get genetic testing. They have to be willing to be monitored. And so due to the slow enrollment, we haven't seen any kind of interim analyses yet. It's still actively recruiting. So if you know someone who might be a willing candidate, refer them to the center. The sponsor is willing to support travel to the sites to do that.
Can you just give them a brief rundown of what ATLAS is?
I'm sorry, ATLAS is the actual formal clinical trial monitoring pre-symptomatic individuals that carry the SOD1 mutation.
Okay, just SOD1, right?
Yes, just SOD1.
Okay, I'm gonna flip the page here and go to the two gentlemen on the panel. First question is, do patients need to be trached to get the brain implant?
No, you can have a trach, you can be on a ventilator. which some of the applicants, some of the implantees have. You do not need to be trached. What you would need if you do not have a tracheostomy is you do need to have a fairly good lung function, because the procedure itself involves lying flat, and it involves general anesthesia, which means anyone who gets surgery with general anesthesia is intubated. If you don't have good lung function, it's very difficult to then remove you off the breathing machine after the procedure. So there is a criteria for having at least 50%, if that number means anything to people, 50% of predicted forced vital capacity to participate in the study, or tracheostomy.
Okay, so I think that kind of answers this next question. How early in the disease can BCI be done?
That's a really good and difficult question. It's a matter of risk and benefit. So if we had BCIs that could, master the universe, then, we would do it earlier. But right now we're still in the early stages. So when you take risk versus benefit, we want to be able to provide alternate ways of controlling a smartphone, alternate ways of controlling a computer, but it's not as fast as, frankly, you can do it without ALS. The benefit would not warrant someone with pre-symptomatic ALS getting a brain-computer interface. You have to have severe paralysis, severe dysfunction of your hands to even qualify for this study.
Okay, next question. How do you actually control devices through BCI? Are you thinking commands?
Maybe Kurt, do you want to?
Yeah, so great question. And so right now, what we're doing is we're reading motor signals, or we're translating those motor signals into essentially a click, a zoom, a scroll. So that's how we're doing it first. So it's not thinking speech or a sentence. It's not thinking I want to shop or buy that. So we're doing it through that understanding of motor function, whether it's moving your hands or thinking of moving your feet. We're taking that and then translating that into, again, a digital motor output.
Sounds kind of like the first eye gaze devices we had like 20 years ago. And it was just a click on like a very long time to type out one word, but it was the eyes watching for that one letter to come across. So it kind of is like the opposite, but the same. Here's a question in chat. Another question: cost of the stent trade versus cost of the non-invasive synchronized stimulation to increase nerve transmission for hand muscles.
Yeah, I'll start with the BCI. So as of right now, the cost through the trial is, we obviously pay for the cost of the trial, that the trial covers the cost. Again, we're working out what the ultimate price, and certainly working with CMS to ensure that these type of devices can be covered. That's important for us and for patients. So that's something that we're working right now with CMS,
Medicare,
the government.
Medicare.
Yeah, Medicare. Yeah.
Can you repeat the second part of that about the?
Oh, sure. Let me get in there. Okay, so versus the cost of non-invasive synchronized stimulation to increase nerve transmission for hand muscles.
Right. So for now, of course, participation in that study doesn't cost anything. In fact, you get a small stipend for coming in. Eventually, if this works, we would use commercially available devices that people with spinal cord injury are very familiar with, that provide kind of electrical stimulation over the body, but it would have to be combined with a magnet over the brain. So I can only estimate it would be in conjunction with companies to commercialize that technology. It would be several $1,000s, but just like Kurt said, all of these approaches are meant to, if they show any benefit, they're meant to hopefully be covered by health insurance in the future. Now, if we only could get health insurance for everyone, that's a separate seminar.
And you don't have to be a veteran to participate in that study, right?
Yeah, thank you. So you do not have to be a veteran to participate in research at the VA. You do not have to be a veteran.
Okay. Any news on identifying prodromal signs in ATLAS?
So at the moment, there aren't definitive signs at the, at present, what we do know and what we've put into the study protocol is the elevation of neurofilament. And then sometimes when you do EMG, there is minimal findings on EMG and clinical exam that could signal early changes. A physician by the name of Dr. Michael Benatar has been doing a lot of research on prodromal states in pre-symptomatic carriers. So the only thing we know is that neurofilament goes up before someone actually develops ALS, but we don't have a definitive answer.
Terrific. Okay, as a person in ALS in my family, I am finally interested in getting genetic testing. Thanks for your talk. I'm in New York City. What are the best next steps?
I guess I'll take that. So I think it's great that you're open to getting genetic testing. The first step is really making sure you're connected with an ALS specialist or an ALS clinician. If you're already connected, I would highly recommend discussing with them directly about getting genetic testing, and they can link you with a genetic counselor or order the genetic testing themselves and provide you the benefits and risks associated with genetic testing. It's really important to have that conversation before you get your testing done. There are laws called GINA laws that help to reduce or prevent discrimination based on genetic information, but it doesn't protect you entirely. So it's really important to have that conversation, and it's different from state to state. So it's important to have that conversation, know what you're getting tested for, know how it would impact your family members should you test positive before you get the results.
Okay, how about back to Dr. Harrell. How long are patients on the anti-clotting medications?
Great. So you start taking aspirin and Plavix 1 week before the procedure. Then you continue both of those medications for 3 months. And then if everything's going well at the 3-month checkup, you continue on aspirin alone and stop the Plavix. But most people will stay on aspirin indefinitely, which a lot of people are on anyway.
Okay. Would you like to see enhanced motor function in humans in general or solely for those who need it, like ALS patients? I think that's on your hand thing.
Oh, that's a very broad question. And of course, there are people out there, you may have heard of Elon Musk. No, but I'm not saying that in a bad way. Yes, these technologies in theory could enhance function for anyone. We're definitely not there yet. And there's a lot of things that could go wrong. So I know I'm speaking for myself. I think I'm speaking for all of us here on this call. We are definitely focused on improving disability first for people with ALS, spinal cord injury, stroke, cerebral palsy, other things. But there's no reason that in theory that these technologies couldn't be applied to try to create superhuman function, frankly. But that's still down the road.
So the $6 million man.
Might get 3 different opinions on that one.
The $6 million man may cost $6 billion at this stage of the game, but Steve Austin. And I'm much older than some of you. Okay, thank you. I think this is for Dr. Andrews. In your opinion, why do you think the list of genes associated with ALS is rising every year?
Yeah, I don't think that there's like these spontaneous gene, although that can happen, de novo mutations do happen, but I think likely we're learning about more and more genes because the technology has gotten much better in genetic screening and genetic testing, the assays and how we do them and the methods. We also, I think, as a community have realized that there is a strong genetic component in certain groups of ALS. And so it starts with people coming to the clinics and clinicians recognizing families that might be involved and then doing the analyses, and genetic counselors being incorporated in the clinic helps us do more testing. In the US, just so you know, genetic testing, the ALS panel, are free. And so that will likely help us learn more about future genes. One thing I will say is that often when you get these tests, sometimes you get what's called a variant of unknown significance. This is a point of major contention because it's very confusing for you, it's very confusing for the clinician, and sometimes it warrants further investigation, and that's why genetic counseling is so important.
Thank you. Okay, what if someone with ALS is on Medicaid? What treatment options are available to them, since Dr. Harrell and Kurt mentioned that they are trying to get insurance coverage, Medicare, for these devices? Anyone?
Okay, so let me start first while Dr. Harrell and Kurt think about things. So when it comes to just medications, approved therapies that are approved by the FDA for the indication of ALS are typically covered in some way by Medicare and Medicaid. I just want to clarify that clinical trial investigational therapies, so experimental therapies that are often offered through clinical trials, are not things that require insurance coverage. And they're usually provided through sponsorship from the manufacturer or the drug company making that drug. So typically the procedures associated in a formal clinical trial, so the evaluation with the doctor, the assessments with breathing and muscle, EKGs, blood draws, are usually covered under the clinical trial grant, not charged to your insurance, not charged to you. And some trials may offer support to travel to the institution, some trials may not. I've been advocating for clinical trials to support transportation to clinical trial centers if it requires in-person evaluations, but that's the story about experimental therapies.
Gentlemen, you have anything to add?
Yeah, my only piece is, at least as far as the BCI world, I don't believe that there's any at this point that are approved, say, through the Medicare, Medicaid process. So, yeah.
Yeah, this is, of course, a huge problem with healthcare in general. No one has solved, the multiple aspects of this problem of financing healthcare in America, for sure. I would dare say there's not really any perfect system in the world, but that's a tough one.
I will say that Medicaid does provide more hands-on care, so the patient gets more intimate care than Medicare does. So Medicare will provide the medication and the equipment, but the help in the home through Medicaid is very beneficial. Okay, how much training is required for the BCI device? Is it similar to eye gaze?
Yeah, I think Dr. Rell answered it well. Usually within the first 1 or 2 sessions, the users are able to control some form of digital environment through the study itself. As you looked at the follow-up over the 12 months period, usually our team goes out once or twice a week to work with the individuals, and we're continuing to kind of push the research, understanding how much we can do with these technologies. So it's definitely kind of that exponential, kind of training element there. And as we talk about, it's a safety trial, but we're learning as we go with each of the patients. And so because each patient is an individual and they control the system, and no patients like the other. So but it happens fairly quickly. Yeah.
So if somebody has a history of ADD, it might take a little bit longer. Right?
Yeah, we don't know any of those variabilities in patients yet, but it'd be interesting to see.
So Dr. Harrell, when you showed us the gentleman using the BCI, you said he was locked in. Can you just explain to some of the people out there what locked in means?
Yeah, thank you. So he has some ability to move his eyes, and he actually can grin, as you saw at the end of that video. But he essentially has no other way to communicate before getting the Stentrode. He had the ability to use the eye gaze device, and he actually uses it pretty well. But locked in essentially means you have no motor capabilities to accomplish any motor tasks. There's a very famous book written by a French author who had a stroke that made him locked in. He slowly communicated by blinking one eye and he had a team of people transcribing his eye blinking into a book about his condition. That's called the diving bell and the butterfly. Anyway, that syndrome is called locked in. So it can happen with ALS, can happen with certain types of stroke, or certain types of cerebral palsy, or MS even.
So historically, I've met patients or known of patients who were no longer able to communicate through the blink of their eye or the gaze. Would it be too late for them to get that BCI?
So,
Kurt is adamant no.
right.
Yeah.
Go ahead.
Yeah, no, I think those are the, I mean, the system works with eye tracker or system, and it just, it alleviates the dwell time essentially with some of these systems if you still want to use it with it. But if they're not able to use the eye tracker, and some of our patients, I believe, lost that ability to use the eye tracking systems. So you can still use the BCI from that standpoint. Yeah.
How would you get consent from that patient?
Yeah, that's the only caveat is for this
Yeah.
study, the person themselves has to consent. So they have to have that ability at least at the beginning of the study.
Mm.
But that video, and yes, certainly the application is to be able to use this without any eye gaze, and it is possible.
And you also mentioned that the patients who are being involved in this study, or the subjects, need to have a companion or a family member to go with them. How much training is involved for the family member?
Yeah, it's a great question. We're learning that as we look at kind of home use. So when our team's not there, how much does the caregiver have to do it? But there's a little bit of training, but most of the time, the use of, as of right now, is our team is there to help out, and eventually they'll start to coach and have the caregiver be able to do the system. But essentially, it's kind of like hooking up a computer, so the task isn't too tough. Yeah.
And we're also, seeing how this affects the caregiver. So the caregivers filling out diaries as part of this study.
I was saying maybe they wanted them to be quiet. Maybe. Okay. So here's a question that came through. It says, loaded, tricky question. Time horizon for BCI commercially being available. Any ideas?
Yeah, great question. I see it obviously always depends on the FDA. So we work with the FDA in our trials. So it's a matter of getting the trials enrolled, and then obviously getting great results, and then working with the FDA to come to commercially. So I do think within the next say 3 to 4 years, we'll be able to have something like this on the market. And again, we're doing our early trial right now that Dr. Ralph spoke about, the COMMAND trial. After that, we'll be moving on to our pivotal trial, which will be a bigger trial that essentially is the trial that will allow us to have a commercial product on the market. But again, all depends on the results in working with the FDA.
All right. Now I'm going to say the ALS Association oftentimes takes equipment from other patients after they pass away, and it reduces the cost of living with ALS for patients. Now, the BCI, you implant something and you implant something, any of this going to be able to be recycled or reused or donated?
Yeah, great question. The implantable portion, no, because that's a permanent implant prosthetic. There is an external piece that will go with this, kind of, it's a size of maybe a little bit bigger than an iPhone that kind of sits and helps communicate that out to the computer. So there is that external piece that would be able to refurbish and repurpose and things like that. So I think that's definitely a possibility. We're not doing it yet, but I think that's definitely a possibility. Absolutely.
Let me just do one look through the Q&A here and see if I have anything else. No, I don't see any. We have thank yous to a few people, to everybody from a few people. So I guess I'll just throw it out there. Last call for questions. Bing, bing, bing on the bell.
I'm seeing a different screen, I guess, but Peter Werner is asking another very good question about.
Okay, go ahead.
He's asking about any exchanges with Lee Hochberg, who's basically the head of the BrainGate collaboration as much as protecting IP allows, of course. Maybe Kurt can take.
Yeah, the answer is what I found is the beauty of the answer is yes, we're working with really Lee and some other folks as well, because I think it's important that we look at this from a community standpoint. If you look at industry, we want to make sure that we even though there's this competitive, there's a lot of neurotech, there's a lot of BCIs that are coming up, certainly, but I think it's a collaborative effort that really starts to bring the best technology for these patients. So yeah, we work with Dr. Hochberg very closely and stuff, and so and some other great kind of BCI pioneers as well.
Okay, so now I've scrolled all the way down and I get to the thank you at the end. So any final comments for any of you?
I see another question. I'm not sure if I have. There's
Well, maybe yours are faster than mine because I just scrolled to the bottom.
Some of yours I haven't seen. I don't know. It's
Okay.
different. But if
If
you
you've already had another stent, can you still have this procedure?
Yes, I think every individual is different, but it depends on and it probably wouldn't be a stent in the particular portion that we're placing the stent showed. That's not a typical area that you'd be stenting. So most likely, yes, especially if it's a cardiac stent of the sort. But I think we got to look at every patient, and if would want more information and a little more clinical look, more than happy to kind of take a deeper discussion around that. But most likely it would be a different location, so you'd be able to do it. Yeah,
not in the brain.
Not in the brain. Yeah.
And there's many other things that are looked at to make sure that vessels are suitable for stenting before it can be safely performed.
That's right. Yeah.
Okay. Now, do you see any more questions?
No.
All right,
Great
good.
questions, though.
All right. And I have no questions on the chat box either. So I will say final comments. Dr. Andrews, any final comments?
No, I think this is just an exciting time, and I really am grateful for the opportunity to speak today and share some of this information. We should do it again soon.
Dr. Harrell, any final comments? Whoops.
I echo Dr. Andrews' comments. This is a great forum, and we'd be happy to try to answer more questions and engage offline as well.
And Kurt, any questions?
Yeah,
Statements.
just thank you for ALS Association, the Greater New York chapter. It's been great to start to meet you, and just really great to see all the technology trials to again try to kind of fight ALS. And so again, if there's any questions on what we present today, more than happy to answer them, or myself or Dr. Harrell. So exciting day. Thank you.
So I just want to say I lost my father to bulbar ALS 30 years ago this year, and he didn't communicate with us for a long time. He was able to move his one index finger left and right. So that was yes and no. So we played 20 questions, more like 120 questions every time he needed something. So and he was a veteran, so he would have been right out there. And this would have been very exciting for our family. So I support you on that. And I just want to let you all know, and thank you very much for your participation, and all of you who stayed on so long for questions and answers. Thank you for your attention to this very exciting program that we had today. Our next session starts at 1:45, and I can't remember the name of it. Oh, it's like, I had it written down somewhere. It's navigating medical decisions and getting the support you need. So clearly, that's something that we all need for our patients as well. Thanks a lot, everyone, for coming out today, and thank the three of you for such a marvelous presentation. Thank you.
Thank you so much. Bye-bye.
Thank you.